Did you know that by simply comparing DNA sequences, scientists can trace evolution, predict protein functions, and even discover disease-causing mutations? This process is called sequence alignment, and it's one of the core techniques in bioinformatics. What is sequence alignment? Sequence alignment means arranging two or more DNA, RNA, or protein sequences in a way that highlights similarities and differences. These similarities tells us about gene function and evolutionary relationships. For example, we have sequence 1 and sequence 2. Matches are highlighted in green and the gap is highlighted in red. As you can see here, most of the sequence is the same, but one small gap appears. That gap represents a mutation, an insertion or deletion that happened during evolution. Pairwise versus multiple alignment. Now alignments can be of two main types, pairwise and multiple. Pairwise alignment, comparing two sequences only. Multiple sequence alignment, MSA, comparing three or more sequences together. For example, we have three sequences, sequence 1, 2, and 3. In this protein example, you can see conserved regions, positions where all sequences have the same amino acids. These conserved areas often indicate crucial functions in the protein. Types of alignment. There are two major strategies for sequence alignment, global and local alignment. Global alignment tries to align entire sequences even if they are different in some parts. And local alignment focuses on finding the best matching regions, not necessarily the full sequence. Scoring system. How do computers decide which alignment is best? They use a scoring system. Match is equal to 1, mismatch -1, gap -2. For example, we have sequence 1 and sequence 2. So the score will be 1. Higher scores mean better alignment. For proteins, special scoring metrics like PAM or BLOSUM are used to reflect evolutionary substitution patterns. Why alignment matters? Alignment is not just theory, it applied everywhere in biology. Evolution, finding common ancestors. Medicine, identifying disease-causing mutations. Drug discovery, comparing protein binding sites. Agriculture, studying crop genomes for resistance genes. Popular tools. To perform alignments, scientists use tools like BLAST, Cluster Omega, and Muscle. These tools can align DNA, RNA, or proteins within seconds, something that will take human years to do manually. So sequence alignment is the foundation of bioinformatics, helping us understand function, evolution, and even disease. In our next video, we will dive deeper into BLAST, the most famous alignment tool.